Welcome everybody to the first of our quarterly acid-base case conferences. I want to thank our wonderful fellow Mohammed Hussein for helping gather the data for this case. So this is a 73-year-old male. In 2016, the patient had a Whipple procedure for cholangiocarcinoma, and for those of you that don't remember what that is, the duodenum is cut out and you attach the biliary tree, the pancreas, and the stomach to the jejunum, essentially. In January of 2021, the patient had a loop colostomy because of a sacral decubitus ulcer. And then in June of that year, the patient developed sepsis and a PEG J-tube was placed. In July, additional sepsis. The patient had a left hemicolectomy, G-tube, and a J-tube. And then in August, the patient developed acute cholangitis with biliary drainage tube placed and gastric biopsies revealed again the cholangiocarcinoma. Patient was discharged on oral feeds and a J-tube with feeds since the G-tube site had closed with bilateral biliary drainage tube. So a very complicated surgical case. Then in September, the patient was readmitted with failure to thrive, abdominal pain, gastrostomy site was draining, and the J-tube was draining brown fluid. On exam, the patient had a left upper quadrant gastric fluid fistula, right lower quadrant J-tube, right flank PBD to biliary drainage bag, a left upper quadrant PBD. A PBD is a percutaneous biliary drainage tube right into the liver to drain the bile. Left lower quadrant colostomy drainage bag, and a suprapubic catheter to biliary drainage bag. And during the hospital course, an NG tube and G tube were placed. The patient continued J tube feeds, and the biliary drains were internalized, and there was replacement of the G tube because the patient developed a gastric outlet obstruction. And then on hospital day 11, the patient, a stroke code was called. The patient had altered mental status. His hands were moving bilaterally, and he had dysarthria. Initially, they thought he had a stroke, but they decided ultimately that it was a metabolic cause. So this is the chemistry panel, and just a few things to note. Everything was quite normal until the 14th. The patient had hypokalemia, which was intermittent. If you look at all these other numbers on subsequent days, the patient was likely getting potassium. And then on the 16th, the total CO2 increased. It was running around 27, 28. It was 31 on the 16th. And if you look at these numbers, subsequently increased, it was 44 on the 20th. That was the peak, but it stayed around 40 until the 22nd, when it fell from 40 back to normal within a day, essentially. And then looking at more lab values here, with the total CO2 that was measured, the calculated bicarbonate, these are all venous blood gases. You can see was very high. On the 21st, the bicarbonate was 51, almost double normal. That was its peak, and remained high until roughly the same time as the total CO2 and the venous numbers came down. And you can see the associated pHs are all high here. Of note, if you look at the PCO2, just as an aside, it's higher than one would expect for a compensation in a metabolic alkalosis. So the patient also had some respiratory, ventilatory abnormalities, was not intubated. And even when the bicarbonate returned close to normal, the PCO2 still intermittently was elevated. So there was associated respiratory acidosis also. And then as far as the therapy, when they noticed the elevation of the total CO2 on the 21st, they gave these solutions, and they also started a proton pump inhibitor. On the 22nd, the solutions were changed. The patient got potassium, half normal saline was switched to normal saline, and this continued to the 23rd. Now, in a metabolic alkalosis, recall that we always try to determine if it's chloride sensitive or resistance. In other words, will the patient respond to chloride or will the patient not? And we divide the diseases that cause a metabolic alkalosis up clinically. And we predict whether the patient will respond to chloride based on what the chloride concentration is in the urine And what the textbooks say is if it's under 15 or 20, the patient will respond. If it's over that, they won't. And this patient had a chloride. There were only 2 urines measured on the 21st when it was 41, and then with subsequent IV chloride therapy, it went up to 114. And I'll discuss these numbers shortly. Here's the complicated input-outputs. Of note, the IV increase, that's this color here on the top, the input. And on the output, you can see various GI outputs, which are a little complicated to see here. So the next slide shows this with a little more clarity. The green is the NG output in milliliters per day. The gray is the drains the patient had, and the blue is the colostomy output. It says stool there. There was only one day where stool was mentioned. It was mostly just colostomy output. And shown below these outputs is the total CO2. And what you can see is the total CO2 notably went up on the 16th to 31 from its previous steady state normal value. And the interesting thing about this graph is that there's no gastric output until later in the course. And so the elevation in the total CO2 appeared to be secondary to the drain output, which had the greatest magnitude of fluid output, and potentially also the colostomy output. Although the colostomy output increased even further. On September 22nd was the day that the total CO2 decreased from 40 to 29, secondary to all the IV therapy the patient got. So the patient looks like he had a chloride sensitive metabolic alkalosis, and then the total CO2 normalized essentially and stayed there until discharge. Now, in the GI tract, there's 2 organs that can lead to a metabolic alkalosis. On the left is shown the transport model of the stomach with the lumen on the left and the blood on the right. And in the lumen, there's a potassium hydrogen ATPase that sends a proton into the lumen of the stomach in exchange for chloride. That proton came from carbonic acid, and so a bicarbonate is generated within the cell, and the bicarbonate leaves into the blood side on another transporter called AE2, anion exchanger 2, in exchange for chloride. Chloride that gets in just leaves into the lumen on this chloride channel. So this is the cause of why we get a bicarbonate load from the stomach after eating. It's called the alkaline tide, and if it's excessive from vomiting, we can also generate a lot of bicarbonate this way, which gets into our blood. The second organ in the intestine that can lead to a bicarbonate load is the colon. The transport processes are different. This is the luminal side up here. And there's 2 proteins here that are required for that to occur. One is called NHE3, which transports 1 sodium ion from the lumen of the colon into the cell in exchange for a proton. And it's coupled to this other transporter called DRA, which is a chloride bicarbonate exchanger. And this is the transporter that's mutated in an entity called congenital chloroduria, which is associated with a metabolic alkalosis. Now, unlike the stomach, one gets a bicarbonate load into the bloodstream when this transporter isn't working. So when it's normally working, bicarbonate is secreted in the lumen, and in effect, you absorb sodium with a chloride because the proton and the bicarbonate just nullify each other. But when this thing is mutated, or in other situations where you can have bacteria inflaming the intestinal wall, or an ulcerative colitis, you can actually inhibit the transport of this transporter, and that impedes bicarbonate secretion into the lumen, and you can end up with a bicarbonate load. Even though in general, intestinal diarrhea leads to a metabolic alkalosis, it's known that colonic diarrheas, especially if they impede this transporter, can lead to a metabolic alkalosis. So what are the interesting features of this case? First of all, the severity of the metabolic alkalosis seemed to be out of keeping with what was happening clinically. In addition, the BUN and creatinine didn't go up. There was no real evidence of volume depletion. They stayed essentially the same throughout the course. Typically, in a chloride-sensitive metabolic alkalosis, we tend to get pre-renal failure. The hypokalemia was intermittent, although there's no data on K balance. It wasn't measured in any of the I's and O's, there was hypokalemia, but not constant. As far as the blood gases, the venous blood gas bicarbonate, which has to be chemically always less than the venous total CO2, was in general higher, and there's just no clear explanation for why that was. And then on the 21st, when the total CO2 was close to its peak, the urine chloride was measured, and it was initially 41. It's not as low as we would expect. The urine concentration can't account for it. Sometimes you can have a patient with a chloride-sensitive metabolic alkalosis, and the urine chloride isn't as low as you would expect because the urine's very concentrated. The osmolality was in the 300s, so it doesn't account for it. But based on this spot urine, assuming the urine volume for that day, we can calculate that about 63 milliequivalents of chloride was coming out that day, and the input was around 462. So clearly, even though the concentration might not have been as low as we would expect, the urine chloride excretion rate was very low compared to the input. We don't have chloride losses from all the other outputs, so we can't calculate if the urine excretion was appropriate or not. We can't do I's and O's of chloride for every solution, unfortunately. And then on the 23rd, the blood total CO2 had decreased to 28. The urine chloride was 114. And using that spot urine and the urine volume, you can calculate an estimated excretion rate of 361, which is what you typically see in a chloride-sensitive metabolic alkalosis. When you're giving the chloride, you don't see chloride in the urine until the total CO2 is corrected. Then the chloride comes out, and we'll discuss it in much greater detail. So as far as metabolic alkalosis, this is the most complex acid-base disorder pathophysiologically to understand. And insights into the specific molecular transport mechanisms in the nephron have made it possible to address the reasons for the phenomenology observed at the clinical level. And despite a literature that dates back to the 1930s, at least, there are still unresolved issues that are controversial. So what are the blood chemistry changes? This is normal blood chemistry. And what happens is the bicarbonate goes up and the chloride goes down. And the anion gap also goes up a little bit because of the elevated pH. You deprotonate albumin and you increase the charge on albumin. So even though the albumin concentration is the same, the charge contributed by albumin to the anion gap goes up. So you get these kind of interesting effects. There's also a respiratory compensation, which everyone forgets, not just in metabolic alkalosis, acidosis, and there have been a number of different studies shown here, but the average slope is about 0.7. So for every 10 increase in the total CO2 or the bicarbonate, the PCO2 goes up about 7. People get different slopes, but that's the average. And when we try to predict the therapeutic response to chloride, we assess most often the urine chloride, but far better than that would be the fractional excretion of chloride or a 24-hour urine chloride excretion rate, because then you're not dependent on your water intake, which can affect the chloride concentration. Same sort of thinking as when you're assessing the urine sodium. It's far better to get a fractional excretion. So over the number of years since it was first described, there's a whole slew of hypothesized factors involved in metabolic alkalosis, and every clinician has their favorite factor. There's hypokalemia, as distinguished from total body K depletion. There's ECF volume changes, aldosterone, collecting duct luminal sodium concentration, or collecting duct luminal chloride concentration, whole body chloride depletion, and even the GFR. And we'll discuss all those in the literature involved. Metabolic alkalosis has 3 phases that must be understood clinically. There's the so-called generation phase, the maintenance phase, and the correction phase. So the generation phase is how it was created, or another way of saying it is, why is the bicarbonate elevated? The maintenance phase tries to address, well, you have an increased blood bicarbonate concentration, but why isn't the kidney getting rid of it? And then the correction phase is the phase that nullifies the maintenance. You did something to stop the maintenance. And again, all these factors have been suggested to be involved in the generation, maintenance, and the correction phases, making the understanding of the pathophysiology, the maintenance, and the correction very complicated. But just conceptually, if you think of a very simplistic mechanism of how the blood bicarbonate can can increase, so-called generation phase, it really can only increase in a general sense by two mechanisms. Either there was a source of bicarbonate, so the amount of bicarbonate in a given volume of water increases, as shown here, or you lost water around the same amount of bicarbonate, and so the concentration went up, and that's been called a contraction alkalosis, which I'll talk about. Now, just globally, talking about the mechanisms for lack of bicarbonate excretion, the so-called maintenance phase, the interesting phenomenon is that the normal kidney does not allow the bicarbonate to increase above 24, thereby preventing any maintenance. And this is data from a rat from 1969, where you can see that as you increase the plasma bicarbonate concentration, above 24, if you look at this dotted line, you start getting excretion. And that is exacerbated by volume expanding the rat. But the whole phenomenology starts to occur around 24. So above 24, your kidney just starts excreting bicarbonate, not allowing the blood concentration to go up any higher. As far as the generation phase in more detail, these are all the sources of where bicarbonate could be coming from. It could be an exogenous bicarbonate load, or it could be citrate, acetate, or lactate, these carboxylic acids that are converted into bicarbonate, where the load exceeds the ability of the kidney to excrete the load. Or it could be an endogenous source, which can occur from various cells, and I'll talk about this in detail because it's typically ignored clinically, where you get a cellular bicarbonate shift, in essence, from enhanced potassium proton exchange at the cell membrane. Or it could be an endogenous source, such as in ketoacidosis from the liver, where you have ketone bodies sent into the blood, or the muscle and the lactic acidosis. Remember, the ketone body and the lactate are converted into bicarbonate. It's the proton that comes with them that makes the problem. But the ketone body and the lactate are actually good things as far as generating bicarbonate, if they're metabolized. It also could be a renal endogenous source with so-called increased renal generation of new bicarbonate, and I'll remind you of what that involves. This is not bicarbonate that's been reabsorbed. It's new bicarbonate the kidney is generating, and it occurs in the proximal tubule, the connecting tubule, and the cortical collecting duct. Another phenomenon that occurs is that's due to the kidney is the connecting tubule and cortical collecting duct actually secrete bicarbonate. And it could be that you have less bicarbonate secretion than normal, thereby causing the bicarbonate to go up. It also could be the gastrointestinal tract, as I spoke about, where it could be a bicarbonate load from the stomach or the colon. And then we're left also with the so-called contraction alkalosis, where you lose just pure water around the same amount of bicarbonate. So there's no bicarbonate added to the system. You have the same amount, it just finds itself in less water. So as far as the maintenance phase consideration, again, the cutoff seems to be about 24, 25 millimolar. Remember, with a GFR of 144 liters a day, we're filtering about 3.6 moles of bicarbonate. It's a little bit less than a box of Arm & Hammer baking soda. It's a lot of bicarbonate, and yet we only excrete a fraction of that, 10 to 20 milliequivalents per day. So essentially, all the filtered bicarbonate load is reabsorbed in the nephron. And if you shut off bicarbonate reabsorption, you can calculate that in about 4 hours, your blood bicarbonate will be zero. So it's essential that the nephron has transport machinery to reclaim the filtered load of bicarbonate. Now, as far as the maintenance phase, as there's multiple potential changes in nephron function. It could be that the filtered load through the glomerulus exceeds the ability of the kidney to excrete bicarbonate, just because of the magnitude of the elevation in the blood level. But it also could be a decreased GFR. If the GFR decreases, remember there's less bicarbonate coming into the nephron. And so when you have a load, if you have a decreased GFR, you're not going to excrete the bicarbonate as easily. So people with AKI or ESRD, if they get a bicarbonate load, the bicarbonate just stays around in the blood. It doesn't get excreted. It also could be that there's increased bicarbonate absorption in the nephron, either in the proximal tubule, the thick ascending limb, the connecting tubule, or the cortical collecting duct. And as I mentioned before, there could be decreased bicarbonate secretion in the connecting tubule or the cortical collecting duct. So let's now go to some of these factors. So starting off with potassium, depletion of potassium is one of the pathophysiologic factors frequently considered by clinicians when they discuss metabolic alkalosis. The pathogenic mechanisms that might be involved have been analyzed over the years in animals and humans for many decades. But several questions arise. Can potassium depletion cause or sustain a metabolic alkalosis without hypermineralocorticoidism, without depletion of body chloride, without ECF volume depletion, or without a low GFR? And then there's been conflicting results in the literature showing that potassium depletion produced by dietary K deprivation results either in no change in the plasma bicarbonate concentration, or a modest but statistically insignificant increase, or even a slight decrease. In rats, people have described a frank metabolic alkalosis, but again, not everybody has seen that. And in dogs, they uniformly get a metabolic acidosis when you deplete them of potassium. So it's very confusing. So as far as what we should think of in humans, or in dogs, excuse me, the first dog experiments were reported by Renal in 1974. And what he did was, there was dietary K deprivation, and as I mentioned, the dogs got a metabolic acidosis, and he thought it was due to decreased net acid excretion due to the hypokalemia-induced suppression of aldosterone. As far as net acid excretion, I just want to refresh your memory. So remember that the kidney actually generates new bicarbonate. This is not reclaimed bicarbonate. The liver makes about 50 milliequivalents of protons a day from our diet, and the kidney generates around 50 milliequivalents of new bicarbonate to match that proton load. For instance, in renal failure, that's why we get a metabolic acidosis. It's because the kidney can't generate new bicarbonate normally. They combine with each other and make carbon dioxide, which the lung just excretes. So, but the important conceptual point is that this new bicarbonate generation is also equal to what we call net acid excretion. So you can look at it either way. You can look at it either as protons that you're getting rid of, or new bicarbonate that you're generating. You're really saying the same thing. But quantitatively, it's equal to the ammonia excretion plus titratable acid excretion, which is just protonated phosphate minus the bicarbonate excretion. That's how we calculate this 50. So going back, what they found was that the net acid excretion during the K deprivation phase actually decreased. These white bars here below the line, that's less than normal, leading to a drop in the blood bicarbonate concentration. Now, Sebastian in 1982 actually defined the role in humans. There were many other studies, but this was the best one. And what he did was a dietary, so the question is, how do you make dietary K depletion? You either have to, you can't remove potassium alone because it's charged. So you either have to have a swap with sodium, or you have to remove potassium chloride. So what he did was a potassium phosphate for sodium phosphate swap. And this was the first conclusive evidence that moderate K depletion can produce a very mild metabolic alkalosis, but they concluded the effect wasn't important. So what you see here is in the K deprivation period, the bicarbonate went up by 2. There was a decrease in net acid excretion, which occurred in the dog, and that's sort of paradoxical because a decrease in proton excretion you would think would lead to a metabolic acidosis, which I'll talk about in a minute. So this can't be the reason for the elevation in the blood bicarbonate concentration. Associated with that was a decrease in chloride excretion. So the patients went in actually to positive chloride balance. The plasma chloride actually went up a little bit. The body weight went up. Presumably the patients became a little volume expanded despite the metabolic alkalosis. And they looked at the creatinine clearance, and with K depletion, it was actually higher than normal. And then the aldo, as one might expect, was lower. So one couldn't attribute the elevation of the bicarbonate to AKI or aldo in these studies. And in fact, the patients looked a little volume expanded. So the take-home lessons from this, in humans on a normal sodium chloride diet, the change in bicarbonate due to dietary K deprivation alone is very minimal. And the increase in bicarbonate occurred despite a drop in the net acid excretion, which actually contributed to some of the lowering in the bicarbonate. But it assumes that the production rate of protons was constant, and that wasn't measured. The increase in the bicarbonate concentration occurred despite an increase in the creatinine clearance, which would increase the filtered load. and lower the bicarbonate in the blood, a decrease in aldosterone, positive chloride balance, and an increase in body weight, suggesting that the patients were volume expanded. So the question becomes, what's the source of the additional bicarbonate? It's not proton input and output, and we'll talk about that in a minute. Then they went on to look at dietary sodium chloride as a determinant of how high the bicarbonate might go. And what they did is they studied two groups of patients, one on a normal sodium chloride intake and one on a low sodium chloride intake with the same K deprivation. They found that in both groups, the dark circles is the low sodium chloride group. The K deprivation was essentially the same. It looks like it's a bit of a difference, but there really wasn't anything statistical. And the blood K concentration fell to about 3 in both groups. But what they found was the following. The dark circles are the low sodium chloride group. They found in those subjects, the bicarbonate went a lot higher as opposed to the normal sodium chloride intake. And at any cumulative potassium deficit, so this is whole body K input output deficit added together, even at the same cumulative K deficit level, the bicarbonate was higher in the low sodium chloride group. And interestingly, the net acid excretion was different. On the low sodium chloride diet, they actually increased their net acid excretion rate as opposed to the decrease. So the question is, was this increase in net acid excretion or new bicarbonate generation sufficient to account for the elevation of the bicarbonate? And we're going to go through some calculations and numbers now shortly. As far as the chloride excretion, in the low sodium chloride group, as one would expect, there was not much of a change in the chloride balance. Only 28 milliequivalents were gained, as opposed to the normal dietary sodium chloride group, where much more chloride was gained. So again, both groups got a little positive chloride balance in these kind of studies. So the take-home lessons in this study: first, in humans during dietary K deprivation, on a normal sodium chloride diet, the metabolic alkalosis is mild. On a low sodium chloride diet, they get a substantial degree of metabolic alkalosis, and the severity correlated with the severity of the K deficit. Now, since the net acid excretion increased in the low sodium chloride group, the kidney was in part responsible for adding new bicarbonate to the extracellular fluid. Was it the only source of bicarbonate? We'll talk about that next. What are the other possible modulating factors that cause the bicarbonate to be higher? Well, urine chloride concentration. It was low in the 0.48 milliequivalents per liter in the low sodium chloride group versus 51 in the high. The body chloride content was not as high in the low sodium chloride group, 26 versus plus 470. The aldosterone tended to be a little bit higher in the low sodium chloride group. Maybe that contributed. And the GFR, or the creatinine clearance, was a little bit lower in the low sodium chloride group. So maybe that contributed by lowering the filtered load a little bit. Now, let's get back to what is the additional source of bicarbonate. And you can think of this as a bicarbonate cellular shift. So when you get hypokalemic for, let's say, dietary K deprivation or any other reason, you're going to get K shifting out of the cells into your ECF because of the potassium gradient that you've now created. And remember, potassium is the most prevalent cation in the cells. Now, it doesn't shift alone. It shifts in exchange for protons. There's potassium proton exchangers. And when that occurs, the proton going into the cell actually generates one bicarbonate ion in the ECF. So this process is actually a way of, you can think of it, of transferring bicarbonate from the cell into the ECF. The ECF gets a bicarbonate load, and the cell has less bicarbonate. So you can also think of it like the cell getting a metabolic acidosis while the ECF gets a metabolic alkalosis. Now, this process will occur less to the extent that potassium and sodium are exchanged, which can also occur. Now, we can calculate the cellular bicarbonate shift by asking how much total bicarbonate was added to the, first you have to find out the total. So in order to do this, you first have to say how much total bicarbonate was added to the apparent bicarbonate space. Now, there's an equation for this that many of you know that we use often in metabolic acidosis to calculate how much bicarbonate to give. And Fernandez in 1989 derives that equation, discusses this at length. But this is the equation. And so you can calculate if you know, like in this case, we know the change in bicarbonate, we know the initial and final, we can actually calculate how much bicarbonate was added to get the final bicarbonate that we got. And in the low sodium chloride group, you actually can calculate that 203.4 milliequivalents must have been added on average to those patients for the bicarbonate decrease from 23.5 to 31. And in the high sodium chloride group, only 72 was added. Now, to calculate the shift, you take the total added, which I just showed you, and you subtract from that the component that the kidney might have done due to a change in its cumulative H balance. So to calculate the external cumulative H balance, you take the total daily H input minus the output, and you get the cumulative change over the number of days you're looking at. Well, it turns out in the low sodium chloride group, when they did this, the H out was greater than the H in cumulatively of about 8 milliequivalents. So almost in balance, but that would represent 8 milliequivalents of bicarbonate added to the patients on average by the kidney. In the high sodium chloride group, the H out was less than the H in, and so you actually have H added to the body or consumption of 169 milliequivalents of bicarbonate. So to calculate the bicarbonate added from the cells, you take the total minus this, and you end up in the low sodium chloride group, about 195.4 milliequivalents of bicarbonate was added from the cells versus even more from the cells in the high. It's just that the kidney resulted in the consumption of most of that bicarbonate. So you can conclude from this in the Sebastian papers that cellular bicarbonate shifting was the predominant mechanism for where the bicarbonate came from in the low sodium chloride group. And this just shows it pictorially. In the low sodium chloride patients, they had a net of 203 added, 195 coming from the cells and 8 coming from the discrepancy between the proton input-output, the kidneys. Whereas in the high sodium chloride group, they had a net of 72 added because even though more came from the cells, they had a lot more bicarbonate consumed because they were not in proton balance. Their input exceeded their output by 169. Now, the mechanism for this is not clear. This is just the overall phenomenology, but the interesting point is that something we never think of, the cells actually shifting bicarbonate from the ICF to the ECF were actually the cause of the generation. Now, what about the role of mineralocorticoids, which always comes up? And this was a nice study by Kazir in 1970, where they gave a milligram of D-aldosterone daily and then looked at the bicarbonate. And they found that it went up very little. The bottom line is that patients with primary hyperaldosteronism can sometimes have a metabolic alkalosis, but many patients do not. And it's also proved difficult to produce more than a mild metabolic alkalosis in humans given mineralocorticoids. Now, they went on to look at the literature. Primary aldosteronism was first described in 1955, and they had looked at all the patients described in the literature. There were 11 patients that they found until the time of their paper. And what they found was that the severity of the metabolic alkalosis correlated with the plasma potassium level. So it seemed to them, and they hypothesized, that you could get a metabolic alkalosis, but it depended on what your potassium was. If your potassium was closer to normal, it would be mild, and if it was lower, it would be more severe. Holter in 1978 went on to look at this in a little more detail, published a paper where he looked at K deprivation in dogs and its associated effect with mineralocorticoids. Again, like Sebastian, they did a potassium for sodium phosphate swap, and you can see here and they gave DOCA. On a normal dietary potassium, the controls, the bicarbonate didn't do much. But with the dogs that had hypokalemia, it went up more. And what you see here is that the urine pH went down more, so there seemed to be a stimulation of proton secretion. The titratable acid, which is the protonated phosphate, excretion went up. the ammonium excretion went up, and the calculated total net acid excretion went up a lot. And they hypothesized that it was due to the kidney per se generating more bicarbonate. Remember, if the net acid excretion goes up, that's the same thing as saying there's more new bicarbonate generated. And they didn't talk about cellular bicarbonate generation. Remember that even earlier than that, in the 1950s, Relman and also Don Selden had shown that the mineralocorticoid effect is modulated by the sodium chloride intake. If you give DOCA and you have dietary chloride restriction, you will not elevate the bicarbonate, and nor will you actually affect the potassium. It requires sodium getting into the collecting duct, which I'll discuss in more detail. So the take-home points are mineralocorticoids per se, the effect is mild or absent. It varies inversely with the magnitude of K depletion, and it decreases with decreased sodium chloride intake. Now, in the 1960s, Schwartz and his colleagues did a whole series of papers focusing on the role of chloride, not potassium. And what they did was they studied dogs in one of the early papers. They gave DOCA and sodium bicarbonate to generate the metabolic alkalosis. And then they looked at correction with potassium bicarbonate, sodium chloride, potassium chloride, and a chloride phosphate swap. So just looking at this, what you can see is here's the bicarbonate going up. K bicarbonate maybe corrected it a little bit. But when they gave sodium chloride, it corrected. Here, same sort of thing. When they gave potassium chloride, it corrected. And here, when they gave the chloride phosphate swap, it corrected. So the largest fall was when they gave chloride. Didn't seem to matter whether it was sodium or potassium given with the chloride. And they hypothesized that chloride is what's important, not potassium or sodium. In humans, they had a different protocol. They gave a low sodium chloride diet. They gave sodium nitrate, which is known to cause a metabolic alkalosis. And then they gave K-bicarb, K-phos, sodium chloride, or KCl. And what they found, here's showing the effect of KCl. What they found was if they gave KCl or sodium chloride, they could correct the metabolic alkalosis. And interestingly, when they gave the sodium chloride, they could also correct the K deficit that occurred. So the patient, went into positive K balance. So they interpreted again the chloride's responsible, like in the dogs, and sodium chloride and KCl both corrected the K and chloride deficits. But if you think about it, they didn't rule out an effect of potassium still, even though they focused on the chloride in correcting the acid-base disorder. They also did other studies looking at the role of chloride. This was a gastric aspiration study in humans. They did replace the sodium, potassium, and water losses. What they found, and you can see here, here's the generation of the metabolic alkalosis. And what they found is the sodium balance was pretty neutral over the number of days they studied. The patients went into negative K balance and negative chloride balance. The K balance was not from the loss of K in the stomach, even though the chloride balance was negative because of that. The K balance is because when you have loss of gastric contents, the kidney actually starts excreting a lot more K. We'll talk about that in a minute, but you can see here how a ton of K came out in the urine during the gastric aspiration. But potassium chloride corrected all the abnormalities. Again, they attributed the effect to chloride, not potassium. Then they started thinking, well, they have to look at the effect of chloride versus K. Again, the same model, gastric aspiration, replacing the losses. They also had the subjects on a low sodium, potassium, and chloride diet. And this time the correction was just with PO sodium chloride. What they found was the sodium chloride corrected the acid-base disorder, but it didn't correct the negative K balance. So even though the bicarbonate fell to normal and the chloride balance became very positive, the subjects were still in negative K balance, and yet the bicarbonate came down to normal. So they said that this shows more definitively that even if you don't correct the negative K balance, you can correct the acid-base disorder. It's got to be due to chloride. This study's been critiqued because they gave a ton of chloride. And the literature, some people feel, well, maybe they just expanded the ECF volume so greatly that that allowed the kidney to dump the bicarbonate and correct the metabolic alkalosis. Now, what about the volume terminology? We've talked about potassium and chloride, but volume is also discussed clinically. What does the term contraction alkalosis refer to? Does it simply mean volume depletion and therefore have a role in the maintenance phase? Does contraction alkalosis refer to the generation of phase via loss of water around a fixed amount of bicarbonate? What is the role of volume depletion? A low GFR and therefore a lowered filtered load of bicarbonate in the maintenance phase. And conversely, what's the role of volume expansion and correction of a low GFR and the filtered bicarbonate load in the correction phase? Well, contraction alkalosis was first looked at in 1965 by Cannon. And what they did was they gave humans ethacrynic acid, and they interpreted their results to show that the bicarbonate went up in the blood, as you see here, and everything else as you would expect in a metabolic alkalosis. But it occurred because there wasn't bicarbonate added, but that water was lost around a fixed amount of bicarbonate. And they called it a contraction alkalosis. The role of contraction has since been looked at. This is a nice editorial written in the mid-70s, and it's currently thought not to be a factor clinically. One, the water loss mathematically is not great enough. And secondly, what tends to be ignored is that there are non-bicarbonate buffers, and they release protons that consume the bicarbonate. So if you just look at the bicarbonate buffer equilibrium, in equilibrium with other buffers that are lumped here together called B, H, and B, if you concentrate up everything by removing water, everything gets concentrated, and you would think nothing would happen. However, the PCO2 doesn't get concentrated in an open system because you breathe. And so as soon as the PCO2 would start to go up, you would increase your ventilation and it's brought to normal. And what that does is shift the equilibrium. And so everything goes this way, including the release of protons from non-bicarbonate buffers. And when those protons are released, they consume the bicarbonate and minimize the increase that you see here from water loss. So that phenomenon actually minimizes any elevation of bicarbonate you might mathematically think might occur if you just remove water without changing the bicarbonate content. The same argument applies to metabolic acidosis. Expansion of water alone to dilute bicarbonate doesn't lead to a dilution because this results in the reaction going the other way and generating bicarbonate. Jordan Cohen in 1968 looked at volume expansion per se. And what he did was he gave a chloride deficient diet with ethacrynic acid. And what he did was he volume expanded the dogs 3 different ways. He gave a chloride solution that was the same as the chloride in the metabolic alkalosis, so slightly lower. And then he gave a much lower chloride concentrated solution. And then he did the same thing with DOCA. And what he found was no matter what the solution, the acid-base abnormality corrected. So what he interpreted the results to be was that the change in the blood chloride concentration or lack thereof and mineralocorticoids both cannot be factors. And it was the volume expansion per se that shut off the proximal tubule, shifting chloride to what he called a more distal nephron site that somehow magically corrected the acid-base abnormality. He did more studies because he wanted to rule out a change in the filtered load. Maybe the filtered load went up. And so he did the same thing, except he clamped the aorta to keep the GFR to compensate for the volume expansion, keeping the filtered load constant. That's essentially what he did. Even though he gave volume, the filtered load was kept constant by altering the GFR. And what he found was that the filtered bicarbonate load also was not a factor, and that it still appeared that in response to volume, independent of the filter load of bicarbonate or the GFR, there was some shift to the more distal sites in the nephron that altered chloride transport. Now, what about GFR? Let's think about it for a minute. What is the role of a low GFR in the maintenance phase? Remember, the normal filtered bicarbonate load is 25 times 144 liters per day, or around 3,600 milliequivalents of bicarbonate need to be absorbed per day. So let's take an example of a GFR decrease responsible for the maintenance phase. Here you'd have an elevated bicarbonate, decreased GFR, such that the filtered load and reabsorption rate are no different than normal. bicarbonate 35. If we lower the GFR to 103 from 144, our filtered load is still the same. So here you have an example of the GFR lowering totally responsible for the maintenance phase. You don't have to invoke elevated reabsorption because the reabsorption is identical. Let's look at example 2. You elevate the bicarbonate and you have a normal GFR such that the filtered load and reabsorption are increased. So we have bicarb 35, we keep the GFR constant. Now we have an elevated filtered load, so the reabsorption has to increase to 5,000 in order for the maintenance phase to occur. So this is all reabsorption phase going up. What about a combination, GFR decrease with increased reabsorption, both responsible? Well, the bicarbonate's 35, but if we make the GFR 124, so somewhere in between, now we've got a filtered load of 4340 and a reabsorption of 4340. So here's an example where you have modest impairment of the GFR, and the maintenance is due to a drop in the GFR and an increase in reabsorption. So clinically, you can get all three, at least conceptually. Well, the GFR was looked at by Marty Kogan and Tony Sebastian in the mid-'80s. These are studies done in humans, very difficult studies, where they put an NG tube and aspirated the subjects with a low sodium chloride diet. And I won't get into all the details, but what they found was that the GFR drop accounted for 40% of the maintenance and increased reabsorption 60%. Then in the subsequent paper, they thought, well, maybe we didn't really stress the tubule. Maybe if we gave more bicarbonate, we would see a different breakdown. And what they did is they did the same thing, but they gave the subjects sodium and potassium bicarbonate also. And in those studies, what they found was that the decrease in the GFR accounted for all the maintenance. There was no change in the reabsorption that one needed to invoke. Now, in the decade between 83 and 93, Luke looked at all this again. He looked at the stuff Schwartz had done and Kogan and others and Jordan, and felt that they had not sufficiently isolated the role of chloride per se. And what they did was they set out over that decade to do a number of studies to distinguish volume depletion from GFR, from potassium and sodium and all the other effects. And their motto was always the same, essentially. It was a peritoneal dialysis exchange of chloride for bicarbonate to generate the metabolic alkalosis. And they treated this was in rats, they treated the rats with different solutions. In this study, they gave the animals sodium chloride or sodium with a non-chloride salt. And what they found is that the bicarbonate stayed elevated when they gave sodium, but it wasn't with chloride. It only returned to normal with chloride was present. And here they sort of did the converse. They gave a non-sodium salt with no chloride or no sodium with chloride. And they found again that only when they gave chloride did the metabolic alkalosis become ameliorated, whereas when they didn't give chloride, the bicarbonate stayed elevated. This study has been critiqued in that if you look at these hematocrits, they tended to be a bit lower in some groups. They said that they ruled out the hematocrit effect, but they actually didn't. But importantly, it occurred with hypokalemia still present in this group. So the metabolic alkalosis corrected without correcting the K or the K deficit or the sodium deficit that was there. And they coined a new term, chloride depletion alkalosis, saying that this must be due to chloride. So they did additional studies now more chronically. Again, the PD exchange for chloride for bicarbonate, only the group that got chloride corrected. And the correction in this study occurred without a change in blood pressure, GFR, plasma volume, or sodium. And they also ruled out Aldo. It was not required in the correction phase. They then went on to do additional studies comparing albumin to volume expand to zero sodium plus chloride solutions. And what they found is the albumin, even though they significantly volume expanded, the animals did nothing. They had to get chloride. And even in the chloride administered group, they got correction despite ongoing volume depletion and despite an ongoing GFR that wasn't corrected. So they said, again, this is chloride. They went on to do studies in man, again, looking at chloride versus volume. Their model was Lasix, sodium potassium citrate, and a low sodium chloride diet. And they treated the subjects either with potassium chloride or sodium phosphate. It's a bit difficult to see here, but the dashed is here's the creation of the metabolic alkalosis, and this is the correction. The dashed line is the sodium phosphate, no correction, only when they gave potassium chloride. This was a more complicated study. They looked at a number of different balances, but and compared to the sodium phosphate, which I don't have time to get into, but the bottom line was the KCl corrected despite persistent volume depletion, decreased weight, decreased GFR, decreased renal blood flow, elevated albumin, increased renin, and aldosterone. And they interpreted the correction data to show that volume change in GFR, and sodium repletion are not required, and that the chloride deficit and repletion is the key rather than potassium being key and the potassium deficit, which was minor in this case. And they hypothesized that the chloride effect is mediated by distal nephron chloride bicarbonate exchange process. So again, similar hypothesis to Jordan, but a little more refined. They then went on to do micropuncture studies to see what was going on. Where might this distal effect be? And they did again a rat peritoneal dialysis model, and they corrected the metabolic alkalosis with 0 sodium containing solutions, but with potassium and chloride. What they found, micropuncture, you can puncture different parts of the nephron and measure the chloride and total CO2 concentrations and delivery. And what they found was that at this point here, prior to the collecting duct, the delivery of chloride and total CO2 were no different in controls versus their metabolic alkalosis model. So they said whatever is occurring in the correction phase must be after this point, because the proximal tubule, everything before that is transporting chloride and bicarbonate the same. So it must be after this. They didn't define where it is, but they knew that until that point, everything looked normal. Now, things got a little more interesting in 1977 when the phenomenon that the cortical collecting duct actually secretes bicarbonate was first discovered. And this was a study in rabbits, Moberg and McKinney. What they did was they gave acid and alkali loads. The cortical collecting duct is this part of the nephron. What they found is that the rabbits that were given bicarbonate actually secreted bicarbonate. This had never been seen or thought of before. Before that, it was always thought that the nephron's absorbing bicarbonate. In control animals, they got some animals that did, some animals that didn't. When they created a metabolic acidosis with ammonium chloride, all the animals were absorbing bicarbonate. So it looked like, for the first time, this segment can do either secretion or absorption, depending on the acid-base status of the animal. Now, Luke, knowing this phenomenology years later, looked at, again, the model of peritoneal dialysis metabolic alkalosis, and found when he took the tubules out of the animals that had a metabolic alkalosis from the PD, they were secreting bicarbonate. What he also found is that when they lowered the luminal chloride to 0, the secretion stopped. So this is secretion increasing, going down, sort of the opposite. And then when they added the chloride back, the secretion came back to normal. So it looked like the secretion was very dependent on what the luminal chloride concentration was. In fact, in a subsequent paper, they showed that there seemed to be a memory effect, that when they created more metabolic alkalosis, they actually got more secretion. And also the chloride balance, balance in the animals seemed to vary directly with the bicarbonate secretion. So this was sort of a memory effect that when you took the tubule out, it seemed to almost have been remodeled in the animal so that its transport properties changed, even though it was sitting outside the animal. Now, the cause of the secretion was not known until Pendrin was discovered. Now, the gene, this has a little bit of a complicated story. The gene encoding the protein responsible for bicarbonate secretion was found serendipitously. In 1896, Vaughan had reported 2 children with congenital deafness and goiter that we now call Pendrive syndrome. So nothing to do with the kidney. But in Everett in 1997 identified the gene and mutations in the gene that we now call Pendrin. This is the gene name, SLC26A4. Now Scott in 99 identified the function of this protein for the first time and said that it was a chloride-iodide exchanger that is now thought to mediate iodide efflux, not influx, but efflux from the thyroid. In 2001, the kidney story came to be, and in addition to the thyroid and inner ear, it was found that pendrin was expressed on the apical membrane in a subtype of intercalated cells in a segment of the nephron called the cortical collecting duct. And this just shows pendrin in green on the apical membrane of those cells. Beside this is an intercalated cell, we now call the type B intercalated cell, right beside a principal cell that's expressing aquaporin 2 on its apical membrane. Now, Nepper and colleagues were able to create pendrin knockout mice, and what they found here was very interesting. These are now cortical collecting ducts perfused outside the animal that in the wild type secrete bicarbonate as shown by others before, but in the pendrin knockout mice, they were absorbing bicarbonate. They totally prevented the bicarbonate secretion, showing for the first time that the secretion was due to pendrin. So we now know that there are 2 functions of pendrin in the what we call the connecting tubule, and this should be CCD, the cortical collecting duct. Pendrin causes the bicarbonate secretion, but it's a little more complicated than that because pendrin is also responsible for coupled sodium chloride absorption because it's coupled to this other protein called NDCBE, which stands for sodium dependent chloride bicarbonate exchanger. So if we look at the beta intercalated cell just here on the apical membrane, we have pendrin exchanging bicarbonate for chloride. Here it shows it as 2 and 2, but it's coupled to NDCBE often. And when you do the stoichiometry, you end up with nothing happening with bicarbonate because you have carbonate going the other way. The net effect is 1 sodium going into the cell with chloride. This is where we're talking about here in the nephron. This occurs in the connecting tubule and the collecting cortical collecting duct. It's sitting beside this principal cell that I'll talk about in a minute. Now, people then started asking, what's the role of pendrin in mineralocorticoid-induced hypertension and metabolic alkalosis? So what they found in the knockout animal is it had no acid-base abnormality. So this shows here the bicarbonate in the animal. The gray bar here is the knockout. The bicarbonate's about the same. When you give the mineralocorticoid, you do get a discrepancy. So what you see is that the bicarbonate level is actually higher in the knockout mouse, and the thinking was that pendrin is needed to correct the metabolic alkalosis, that in its absence, bicarbonate cannot be secreted, and that's why the blood level is higher. People asked, does pendrin regulate acid-base status in humans? The answer is it's unclear. In 2008, there was a case report of a patient with Pendrin syndrome who got a very, very severe metabolic alkalosis after thiazide treatment. It was corrected with with treatment that wouldn't have expected to create such a degree of metabolic alkalosis. The thinking was, well, this patient can't secrete bicarbonate, and that's why. In 2011, there was another case report of a patient with Pendrin syndrome, again with very severe metabolic alkalosis after vomiting that seemed out of keeping with the vomiting. And then others have looked at pendrin expression in kidney biopsies from patients with distal RTA, and it was found to be decreased. Perhaps pendrin expression going down is ameliorating the RTA. And people have looked at exosomes containing pendrin, which was found to be decreased after ammonium chloride loading and increased after bicarbonate administration. And this just now summarizes what's known. So in metabolic alkalosis, pendrin redistributes from the intracellular to the apical membrane, and the total expression increases. The converse in metabolic acidosis would decreases pendrin expression on the apical membrane. Dietary potassium deprivation decreases pendrin's expression. Dietary chloride deprivation increases it. Dietary chloride loading lowers it. Angiotensin 2 has effects, aldosterone has effects. Interestingly, there's this paracrine system. Alpha-ketoglutarate is one of the important mediators of the Krebs cycle, and is also what is converted into new bicarbonate in the proximal tubule. But it turns out that alpha-ketoglutarate released from the proximal tubule actually binds to this receptor on the type B intercalated cell and stimulates pendrin transport. And I don't have time to talk about this today, but it's an important intratubule signaling system. And then also beta agonists can affect pendrin-mediated chloride bicarbonate exchange and furosemide. So now let's get into the new transport machinery that people now feel is involved in metabolic alkalosis that can potentially explain a lot of the clinical phenomenology. So first, let's talk about ENaC in the connecting tubule and CCD. ENaC is the sodium channel that's responsible for sodium absorption. It's shown here. It's made of 3 subunits. That's what it looks like at the atomic level in a cartoon. But sodium gets absorbed. This is what amiloride inhibits. And this is what is altered in mutations in Little syndrome, where it's acting excessively. But in the principal cell, you have sodium coming in through ENaC that depolarizes the inside of the cell, creating a more favorable electrical gradient for potassium to leave on the ROM-K potassium channel. So that's how ENaC is coupled to potassium secretion, not directly, but electrically. Now, the important point is that when ENaC depolarizes the apical membrane, it also changes another voltage that we call the transcellular voltage and makes the lumen more negative compared to the outside. And that is very important because that drives chloride, what we call paracellularly, between the cells, and it's known to be mediated by claudin-4, a protein that's between these cells. So when you enhance ENaC-mediated sodium transport, you enhance potassium secretion in the lumen and paracellular chloride transport. But there's an additional effect via electrical coupling of this cell with another intercalated cell that we call the type A intercalated cell that's responsible for proton secretion and bicarbonate absorption. When this voltage changes here, there's coupling of the voltage to this cell that enhances H secretion. And that's why ENaC affects K secretion, chloride absorption, and proton secretion, which can lead to a metabolic alkalosis. There's also competition. If the luminal chloride is low, you actually enhance the K secretion and the H secretion. Now, there's another pathway that one has to understand what's going on in the metabolic alkaloses, and that's the 3 pathways for sodium and chloride transport in the distal convoluted tubule, the segment that's just before the connecting tubule and the cortical collecting duct. And you might remember this from last week's talk on hypomagnesemia, and I won't get into the details, but this is the transport machinery in the distal convoluted tubule. Focus here on NCC. That's the protein in the apical membrane that thiazides bind to, and that is mutated in Gittleman syndrome. It's responsible for sodium chloride absorption. It's not a channel, it's a co-transporter, and transports sodium and chloride in equal amounts into the cell. So that's one of the 3 pathways for sodium and chloride transport. The other pathway is the ENaC pathway I talked about, where you have sodium coming in through ENaC, changing the transepithelial voltage, driving chloride paracellularly. That's an electrical coupling, but another way that sodium and chloride can get absorbed. The third way, as I mentioned, is pendrin coupled to NDCBE. But the important point to understand, and also what has great consequences in understanding what's going on at a molecular level in the metabolic alkaloses, is that if aldose stimulates ENaC, you're going to have less chloride available for pendrin and for NCC. And if hypokalemia stimulates NCC, we talked about that last week during the hypomagnesemia talk, it's a very potent stimulus of NCC. You're going to have less chloride available for ENaC and for pendrin. Or if thiazides inhibit NCC, you're going to have more chloride coming down to the ENaC coupled transport and the pendrin coupled transport. If there's less urine chloride because of extrarenal chloride loss, all 3 will be shut down. So there's competition of where the chloride will be transported. So now let's look at each of the original factors that I talked about. Potassium, and this is an involved slide, but this is now what is likely going on. First, you have a cellular bicarbonate shift. The KH exchange process in response to a lower blood potassium leads to K efflux into the extracellular fluid and H influx into the ICF, leading to ECF bicarbonate elevation and a decrease intracellularly. Hypokalemia also, because of this intracellular acidosis, stimulates proximal tubule ammonia production and new bicarbonate generation. It increases proximal tubule, connecting tubule, and CCD-H secretion. And that leads to increased titratable acid formation, new bicarbonate generation, and bicarbonate reabsorption. And because of the intracellular acidosis, there's increased proximal tubule ammonia formation. There's also direct stimulation from hypokalemia in making more ammonia, and excretion, which means more new bicarbonate generation. Hypokalemia also causes pendrin to decrease on the apical membrane, leading to impaired bicarbonate secretion in the connecting tubule and cortical collecting duct. It also stimulates the sodium chloride cotransporter, leading to less connecting tubule and CCD ENaC transport, less K secretion, but also less chloride delivery to pendrin to function and secrete bicarbonate. With the glomerulus, K depletion in the literature has been shown to decrease the GFR, not widely appreciated in man. And its significance in man is not known, but in animals, one can show that, and that would decrease the filtered load of bicarbonate. Correction of K is required to lower the blood bicarbonate. However, if sufficient chloride is given, such as the Schwartz study, pendrin-mediated bicarbonate secretion may potentially be stimulated sufficiently, depending on how severely its expression has been decreased, to correct the increased blood bicarbonate despite concurrent hypokalemia. And then the worse the hypokalemia, the worse the cellular bicarbonate shift you're going to get, the worse the suppression of pendrin, and the worse the bicarbonate secretion, and the greater the stimulation of new bicarbonate generation and reabsorption. What about aldosterone? This is an adrenal adenoma with a chloride-resistant metabolic alkalosis. Well, here we have increased ENaC sodium transport and ROM-K potassium secretion, leading to hypokalemia, enhanced paracellular chloride transport, and therefore sodium and chloride transport is shifted away from NCC in the distal convoluted tubule, and pendrin can't function as well, so there's less bicarbonate secretion. Increased ENaC activity, secondary electrically, will stimulate proton secretion, as I mentioned before, in the type A intercalated cell with increased bicarbonate absorption, net acid excretion, and new bicarbonate generation. Aldosterone does increase pendrin expression, which would be predicted to lead to enhanced secretion, but ENaC-induced enhanced paracellular chloride transport with the competition will decrease the available chloride available for pendrin to even work. So even though there might be more protein on the membrane, it won't work as well. Then aldosterone-induced hypokalemia would all result in the previously described independent effects that I talked about before. But then ultimately, it's a dynamic situation. Hypokalemia will decrease the aldo levels, reversing all these aldo effects. So the net effect at any point in time is complicated and determined by the aldo level, the magnitude of hypokalemia. But again, patients with more severe hypokalemia would be predicted to have a more severe metabolic alkalosis. And although administering chloride has no effect, administering KCl rather than sodium chloride has been shown in this scenario to decrease the blood bicarbonate. What about extrarenal loss of chloride in the gastric alkalosis, where we have a chloride-sensitive metabolic alkalosis? Well, we have a low distal convoluted tubule luminal chloride, which will block NCC, increasing the sodium delivery to the connecting tubule and CCD for ENaC-mediated sodium transport with enhanced K and H secretion. And that means new bicarbonate generation and absorption. The low connecting tubule and CCD luminal chloride lowers ENaC-stimulated paracellular chloride transport, which would shunt some of the luminegativity normally, resulting in even more K and H secretion. The low CNT and CCD luminal chloride will block pendrin bicarbonate secretion. Aldo goes up from volume depletion, which will result in the previous aldo effects. Hypokalemia will result, resulting in the previous hypokalemia independent effects that I talk about. And administering chloride salts will result in an increased luminal chloride in the nephron. The DCT NCC function will return to normal, reclaiming sodium with NCC rather than ENaC. ENaC transport can now be coupled to paracellular chloride transport, and you'll have less K and H secretion electrically. and also in the connecting tubule and CCD, Pendrive can now work and will increase its bicarbonate secretion. And all these things will lead to correction of the metabolic alkalosis. And now lastly, just Barter Syndrome. Volume depletion stimulates aldo. Aldo and increased sodium concentration because of the thick ascending limb impairment will increase the connecting tubule and CCD ENaC transport. Again, that means more KNH secretion, more paracellular chloride transport, leading to hypokalemia with more bicarbonate generation and reabsorption. The aldo going up will result in the previously described aldo effects. The hypokalemia will result in its previously described independent effects. And again, it's dynamic. Hypokalemia will at least reverse the aldo effects when the aldo decreases. So with that, I would stop there. And I know I'm a little over, but I'd be happy to answer any questions that any of you might have.
Ira, quick question. Regarding chloride deficiency, what's the magnitude of effect in terms of chloride bicarbonate secretion versus proton chloride secretion? Which effect is greater? Has anyone looked at the magnitude of those two effects?
Right.
Is one predominant over the other?
Right. So you have to look at it axially too, because it depends. If you're talking about the type A intercalated cell, is that what you're talking about that? Because there's also proton secretion in the intermedullary collecting duct, which I didn't have time to talk about. But if you're comparing, when you lower the luminal chloride, you're actually enhancing potentially proton secretion, right? So if your question is how much is the enhancement of proton secretion versus the decrement in bicarbonate secretion, I don't know of anyone who has looked at that. It also depends. It's complicated because you've got the connecting tubule that's very long with a lot of bicarbonate secretion compared to the bicarbonate absorption in the chordal collecting duct. They're a little more equal. So axially, the relative proportion of the two of them is not the same, but axially you're going to get differences in the chloride concentration depending on the transport, because the previous segment always affects the segment after. So it really depends on what the chloride concentration is in each of the three segments, which are affected by each other axially because the urine's only flowing one way. So the transport in the previous one affects what it is in the latter. They never simultaneously have the same chloride concentration. So
and the other question I have is, when you were discussing about the TM for bicarbonate reabsorption in the proximal tubule, I thought, when you talk about proximal bicarbonate absorption, it seems that you say that it sounds like you say that the TM for bicarbonate is fixed. Is it that volume?
Oh, it's not. No, it's not fixed. I didn't mean to.
It's volume dependent, right?
Yeah, in the first slide I showed the volume dependent. So it definitely is volume dependent.
I signed in late, so maybe I missed that.
Yeah. As you volume expand, you get you get more excretion, and as you volume deplete, yes, no, it's definitely volume dependent. Okay, well, if there are no more questions, thank you very much for listening, and I appreciate the questions also, Mintry. Thanks.